Our today's topic is how genetic information is flowing. How genetic information flows down. This is our today's topic. So genetic information flows by making RNA from DNA and then protein from RNA. Making RNA from DNA is called transcription, and making protein from RNA is called what? Translation. Okay.
Yes, sir.
And this
Yes,
is
sir.
called the central dogma of molecular biology. A particular segment of DNA is called gene, and from this gene an RNA molecule is produced. And this process of producing an RNA from DNA is called transcription. And when this RNA, with the help of ribosome and other components, make protein, then this process is called translation. And this flow of genetic information from DNA to RNA, RNA to protein is called the central dogma of molecular biology. And this is the genetic code. What is genetic code? Genetic code is a triplet of bases, a set of 3 bases which specifies, which encodes a particular amino acid. Such as when there is bases like T, A, C in DNA, then we get methionine in the polypeptide. When CTG is the triplet base, then we get what? Aspartate. So this sort of triplet bases are called genetic codes. There are 20 amino acids, we know. So for 20 amino acids, there are 64 different combination of bases. How 64? As bases are 4, there are 4 types of base, and this genetic code is triplet. That's why 4 cube makes 64 different possible combination of genetic codes. Okay. As for 20 amino acid, 64 different genetic codes. So there are amino acid which are having multiple codons. And this property of having multiple codons for particular amino acid is called codon degeneracy. So there are different type of codon producing, specifying or encoding different amino acid. But what we need to learn by heart is AUG, which is a start codon, UAA, UAG, UGG. These are a stop codon. These 4 codons must be remembered and important for our exam. See, this is a DNA molecule. This is the 5' to 3'. This is 3' to 5' strand. This strand is called sense strand, and DNA is always read in the 5' to 3' direction. So this strand is called sense strand. This is called antisense strand. This sense strand contains triplet of bases, and each triplet encodes a particular amino acid. Now we are going to learn how protein synthesis occurs in details. The first step is the transcription. What is transcription? As we have said earlier, transcription is the producing mRNA from DNA. What is to note that proteins are made on the ribosome. Ribosome is the protein factory. Ribosomes are available in the cytoplasm. You won't find ribosome in the nucleus. Ribosome will be free in cytoplasm, or else, or it may be attached to endoplasmic reticulum. Of course, this is the rough endoplasmic reticulum, but you won't find it in the nucleus. So ribosome, which is the protein synthesizing machinery, is found in the cytoplasm, while DNA is present in the nucleus. So how the message from DNA transferred to the ribosome. DNA must make a molecule which will go to the ribosome and instruct the ribosome to make proteins. So DNA need to produce a molecule which is of course RNA, and when such RNA which contains information for making protein, this type of RNA is called messenger RNA, which carries message for protein. So producing mRNA from the DNA, making the template strand of DNA is called transcription. So how transcription occurs? Let's discuss. So this is the DNA. When transcription starts, the DNA unwinds and unzips as hydrogen bonds between the bases is broken down. Free activated nucleotides come and pair up with the complementary bases, and with the help of RNA polymerase, ultimately mRNA is formed. And when mRNA is formed and gets matured, they leave the nucleus through this nuclear pore. So this is the simple idea how transcription occurs, and that's all about transcription.